Okay, so our next topic is going to be about membrane transporters. Um, and so this is going to be more involving facilitated diffusion or our active transporters. And so here's the objectives to know for this lecture about facilitated diffusion, passive, not requiring energy, as well as our real active transporters. So a simple passive diffusion is basically something that can cross the membrane without any help. It does not need a protein or a channel in order to get across. And so these are going to require no energy, and they're going to typically follow concentration gradients. So oxygen, carbon dioxide, as well as other lipids are going to be able to cross membranes. For other things to get inside, because it's a lipid bilayer, a lot of things that need to get into our cells are going to have hydrophilic residues that cannot cross the membrane, or they're too large. And so they're going to need channels to get through. And so we can have some channels that are going to be uniport, and that means it's going to cross one substrate. Other channels can be a symport, in which two products go in at the same time in the same direction. And then we have our antiport, where two products are going to go in, but one goes in one direction and one goes in the other direction. Our symport and antiport are co-transporters, whereas our uniport is obviously just a single transporter. Now, our fundamental differences between our transporters and our ion channels that we've talked about. So, transporters are going to bind substrates with high specificity. Okay, so they're gonna, whatever is going to take the substrate across the membrane is very specific for that substrate. And they are going to be very similar to enzymes such that they can become saturated. And then they can move a substrate against a concentration gradient, so this is active transport. Now, channels are only passive transport. So channels allow the movement of ions at rates on order of magnitude greater than a typical transporter, and some have specificity for an ion, but they are not saturated. And the direction of the ion is dictated by the ion's charge and the electrochemical gradient. So some facilitated transporters are going to not require any energy. So this is an example of a facilitated transporter, in which you have some substrate that's going to bind, and then the transporter is going to change conformation, which will eventually allow that substrate to go to its inside the cell or outside the cell, whichever has the higher or lower concentration. So this is basically based on concentration gradients. But this substrate itself cannot passively diffuse into the cell, so it has to use this transport system, this protein transport system. And an example for this is the glucose transporter of the red blood cells. So red blood cells have the glucose transporter called GLUT1. And these are highly going to be able to transport glucose at very low concentrations because, as we know, red blood cells need glucose. That's their only fuel. So this is a passive transport. Driven by a concentration gradient. So the red blood cell needs glucose to enter. It's a type 3 integral protein, has 12 hydrophobic segments, and each with a membrane-spanning helix. And here basically is your glucose, and again it binds, and then it gets released into the red blood cell. Now another example of this passive is a chloride bicarbonate exchanger in red blood cells. This is a facilitated diffusion. And in this case, this is an antiport. So in the tissues, you're going to have chloride enter, and you're going to have bicarb exit. And in the lungs, it's the reverse. You're going to have chloride exit and bicarb enter. And it's electroneutral because the bicarb is, and chloride have similar charges. Now, glucose transporters with respect to diabetes, we have GLUT1, which is going to be ubiquitous, meaning it's always expressed, it's always on. It's going to be in the brain, and the red blood cells are going to have many. We have our GLUT2 receptors, which are liver and pancreatic cells, especially those beta cells, some kidney cells in your intestines also. And so these are going to be important in regulation of insulin release from the pancreas and removal of excess glucose from the blood with respect to your liver and your kidney to a smaller extent. GLUT3 receptors are going to be in the brain, and they're also in the testes, and this is similar to our GLUT1 in that they're always going to be on. GLUT4 is important because this is going to actively be activated by insulin. So this is going to be the major GLUT transporter that removes glucose right after a meal. And then GLUT5 is our fructose transporter. So that's important to note. GLUT5 has a higher affinity for fructose. So when you're on a high fructose diet, or if you have those sugars, high fructose sugars in different products, it's thought to be bad. And it's not that fructose is worse than glucose necessarily, but it's because you have now engaged another transporter to bring sugar into your body. So not only are you going to use a transporter for glucose, you're also engaging the GLUT5 high fructose transporter. And so that can increase the amount of sugar actually entering your body. So now let's move on to active transport. So active transport is when you have a solute moving against a concentration gradient or against an electrochemical gradient. So it's basically a movement against a gradient. And so it's basically endergonic, it's very unfavorable. And so it's going to be coupled to an exergonic. So when you have a delta G, let's see, let me do this another way. Let's do a color. A delta G for going down a chemical gradient with a negative charge, it's going to be low. If it has a positive charge, you're going to get transport against a chemical gradient. Now, there are two types we have shown here. There's going to be primary, and then there's going to be secondary transports. And they're both going to be coupled, of course, with ATP to make it exergonic. And so in the primary transport, you're basically going to have the transporter move a substrate out. In the secondary active transport, this is when you have your cell actually needs substance 2, but in order for substance 2 channel to be open, substance 1 has to be released. And so in this case, you're showing substance 1 being released and a co-transport of both substance 1 and substance 2 coming into the cell. And so substance 2 uptake is dependent on substance 1 being present extracellularly for that uptake. So now there's a few type of transporters that we're going to talk about. The first are going to be our P-type ATPases. Our P-type ATPases are going to have a similar structure. They're going to have, again, inside they're going to have an A domain or an actuator domain, and they're going to have a phosphorylation domain and a nucleotide binding domain with the N domain. So you're always going to see an A, a P, and an N domain. You also have the transmembrane domains of T and S. So it's the N domain that is going to bind to ATP. On all of these. Now your P-type ATPases, your N domain is the one that has protein kinase activity, and it's going to phosphorylate the aspartic acid domain or residue. So your N domain is going to phosphorylate. Okay, so it bound to ATP, now it's going to phosphorylate. And it's going to phosphorylate our aspartic acid residues. Now your actuary domain has a protein phosphatase. And what we know about our protein phosphatase is they can remove the phosphorylated groups. So our protein phosphatate will remove the phosphoryl group from aspartic acid residue. And then our T and S domains are going to be changed with the phosphorylation and dephosphorylation. Now, your phosphorylation and dephosphorylation are not going to have any impact on substrate binding to the receptor. So the T domain is going to actually include the ion transporting structure, and the S domain is what's going to provide support for the T domain. Okay, so let's show some examples of these P-type ATPases. Oh, I'll sneeze. Okay, one is the Sarcoplasmic Reticulum calcium pump. So these P-type ATPases can move calcium. We'll see also sodium and potassium in just a moment. But basically, the Sarco pump is going to want to release calcium. And so you have the same sort of setup that we just talked about. You have your A domain, your actuary domain. You have your P domain, phosphorylation domain. And you have your N domain, your nucleotide binding domain. And so the same thing's going to happen is that your ATP is going to bind to the N domain. Your N domain is going to phosphorylate the ASP residues. Your actuary domain is going to dephosphorylate. And all of this is going to allow calcium initially to bind into that channel, the ion channel. and then the calcium will be released from the ion channel, and then the channel will reset itself so it can pick up more calcium. And so all of this changing has to do with affinity. So calcium is initially going to bind, two calciums will bind when the ATP binds, and then phosphorylates that aspartic acid residue. And then in the E2, the calcium has a decreased, the P domain has a decreased affinity for calcium, and so it allows calcium to diffuse away. Another example is our sodium-potassium ATPase. And this is important because sodium-potassium ATPases are maintaining intracellular concentrations of ions. And so this, basically, the movement of sodium and potassium are going to generate a membrane potential. And the membrane potential can be important for muscle contraction, maybe it's to release insulin or other things. But in general, you're going to move 3 sodium out for every potassium you move in, for every 2 potassium, sorry. So 3 sodium out, oops, for every 2 potassium in. Now there are 2 other types, so those were the P types. There's also the V for vesicle type, you can think of that as vesicle type, and the F type, which is very specific to our bacteria, and for us it's our ATP synthase. So for our V type, this is going to be to acidify an intracellular compartment. So for example, our lysosomes. Our lysosomes will have V-type ATPase proton pumps, and that's to maintain a low pH inside of a lysosome. The lysosome digests your cells when your cell needs to break down. Your lysosome will digest bacteria when they enter your cell, and so the low pH is what is required for lysosomes' enzymes to work, and so they have these V-type proton pumps. And so V-type pumps are going to pump hydrogen, so that's different than our P-types. Our P-types pumped calcium or sodium and potassium. And then our F-type are going to catalyze what's called an uphill transmembrane passage of protons, and this is driven by ATP hydrolysis. The F is going to come from the identification of these ATPases as energy coupling factors. Now, the V type is going to maintain your vacuoles or your lysosomes at a pH of about 3 to 6. It's going to be the V0 is going to be the integral transmembrane domain, and the V1 contains the ATP binding site. And then for your F type, you're going to have your transmembrane pathway for protons. And when we talk about the ATP synthase, you're going to see that it's sort of like a crank, and it sort of turns, and protons are going to move from a high concentration to a lower concentration, or to drive the protons uphill into a higher concentration. Sorry, it's the reverse. And so this is a large proton gradient which drives the reaction, which is reverse of that, to make ATP. So this is going to be making ATP. And then our next guy is called our ABC transporters, of which our cystic fibrosis transmembrane conductase receptor is one of them. And remember, this is a chloride channel. So these are large member transporters, and they're going to transport a lot of things. So here I also have another one that is going to be for the transporting of drugs. So we have MDR1 shown here also. So MDR1 is a multi-drug transporter that is part of the ABC transporters. ABC stands for, let me write this out for you, ATP, this is TP, so that's where the A comes from, binding cassette, oops, sorry, binding cassette transporter superfamily. And so the cystic fibrosis transmembrane conductance regulator protein is an ion channel for chloride, as we said, and it's going to be operated by ATP hydrolysis. And this is an ABC transporter without a pumping function. So all others have pumping functions, whereas this is simply a channel. It opens and allows chloride to leave. Now, ABC transformers have two nucleotide binding domains. So it has two binding domains. And so you can see that ATP is going to bind to both of those domains. It has two transmembrane domains. There are 48 genes in humans that encode these ABC transporters. They're important for many things. In addition to our drugs, they also transport alcohol. And so for our cystic fibrosis patients, mutations in this transporter is going to be, the most dominant is going to be a phenylalanine at position 508 is deleted, which is going to cause the misfolding of the proteins, and so the channel actually does not incorporate into the membrane, and so they have a lack of channel. And then finally, our glucose transport. So our glucose transport is going to be a symport, and our glucose transport is going to be co-transported with sodium. So in the intestinal lumen, after you take up a meal, your glucose and sodium are going to come in by a glucose sodium symporter, which is driven by a high extracellular sodium concentration. So coupled with this on the other side over here, is going to be a sodium-potassium transporter. And so the purpose of sodium-potassium transporter is to make sure that in the epithelial cell you have a low concentration of sodium. So you're going to have two potassiums enter for 3 sodiums leaving, and that's going to maintain the gradient for the glucose-sodium transporter to continue. Now, once glucose gets inside of the epithelial cell, it has a glucose uniporter, which is actually GLUT2. It's the same receptor that is going to be found on your liver cells and your pancreas beta cells. So the glucose can use the GLUT2 to be released into the blood so that other cells can pick this up. So again, you have two transporters happening for glucose to be picked up. One is that you have to maintain the sodium gradient, so you have a sodium-potassium pump that pumps potassium in and sodium out of the epithelial cell. And on the other side, on the lumen, you have the glucose-sodium cotransporter, which is a symporter.